Jyota ma'am. Without, I will stick to the time, ma'am.
Yes.
Let me introduce myself. I am Dr. K.L.V. Varaprasad Rao, Assistant Professor of Botany, Government City College, Hyderabad, ma'am. Today, I would like to talk on implantar transformation tomato, which is a high throughput and a novel technique in the transformation. So the plant, briefly, I would like to tell the two methods, which is very highly the successful so far. It is plant agrobacter mediated transformation. It's again the existing method is co-culturing with tissue explant, and my interest of today's topic is implant method. It is also another method. Implant method is the thing which is innovative is you can do the transformation in vivo conditions, ma'am. There is no need of the tissue culture conditions except for the culturing the agrobacterium strain. So coming to the co-culturing with the tissue explants, the co-culture with tissue explants includes, it's the most successful method, as you all know, for the transformation of most of the plant species. And the method involves co-cultivation of the plant explant with agrobacterium and regeneration on a high nutrient medium.
Yeah. Sir, move the slide. Move the slide.
So coming to the
Second one, click change.
historical data, ma'am, it's not moving, ma'am.
Not moving. Moving. Yeah. ఇప్పుడు ఓకే అండి.
Yes, ma'am. Are you going to? Okay. But it is stuck in my system, madam. So that's
okay.
right.
You continue. You continue.
Okay, ma'am. Okay. Okay, ma'am. So అది ఫుల్ కాకుండా మీట్ గూగుల్ మీట్ ఇది అదే ఓకేనా ఓకే మ్యామ్ నో ఇట్ ఇస్ ఓకే మ్యామ్
మీరు కంటిన్యూ అవ్వండి.
It is so these are the Arabidopsis plant, no? The briefly the introduction I will tell you. Arabidopsis is the plant which is the model plant for the transplantation of transformation through implantar transformation also, as it is having the many advantages, as you all know. But our interest is choosing the from the Arabidopsis plant only. We have chosen the tomato plant, ma'am. So here you can see in this picture the Arabidopsis plant, along with it is a cultivation, the co-cultivation with Arabidopsis culture with agrobacterium transformed plantlets which are survived in the hygromycin resistant plant plate also you can see. So coming to the next slide. స్టాప్ షేర్ మళ్ళీ చెప్తాం
పోయింది మళ్ళీ.
ఇది ల్యాప్టాప్ ఇష్యూ విండో
స్టక్ అయిందండి. ఇస్ ఇట్ స్టక్?
అవును మేడం, ఒక్క నిమిషం. సరే అక్కడ ఇక్కడ ఒక్క నిమిషం. అరే రాలేదు. ఓకే
ప్రెజెంట్ చేయండి సార్ మాకు.
Yes, ma'am. So the reason for choosing tomato as a model plant, ma'am, for our studies, as it is the second largest consumed vegetable in India, as you all know, and because of its immense nutritional values, which includes vitamin C and lycopene and beta carotene, and it is genetic model for fruit ripening experiments, and also for studying disease-resistant plant varieties, ma'am. And it is one of the smallest genome among the students members. That is also one of the advantages which we can obtain while doing the transformation experiments. And it is having a relatively short life. That is also an advantage for research, so that we can get more number of generations in a short span of time. And also the extensive germplasm and mutant collection is already available. So all these are advantages that led to the choosing the tomato plant as a model plant for the in-plant transformation studies. So the objective is for the development of an efficient in-plant transformation methodology for tomato, and for that we have taken the plant material, the variety of Arcavica seeds of tomato variety, which is a wild type, obtained from AHR Bangalore and also TGRC. And the agrobacterium strain, the vector we used was Cambia P1303. In this study we have used, and this is coming to the vector construct. So this is the vector construct, ma'am. So which is having a agromycin. It is having the agromycin for the as a bacterial selectable marker, and kanamycin as plant selectable marker. Here, the green fluorescent protein is the reporter gene, so which will indicate the presence of the transgene. By the presence of reporter gene, we can identify that. Also, we can study. So the two methods, the methodology that are obtained, that are adapted for the study is one is floral dip, and another one is floral injection. So in the greenhouse itself, we can take the agrobacterium the culture with our gene of interest, and the entire cluster of the unopened flower buds will be targeted, where they will be dipped in the sucrose solution along with the surfactant, which contains agrobacterium with a OD of 0.6 for 30 seconds to 45 seconds. That method is called as floral dip method. And another method is the floral injection method, where we will use the unopened flower buds. They will be targeted, and they will be emasculated and injected with a sucrose solution along with the surfactant containing agrobacterium culture, which contains gene of our interest. So whether the transformation has been done or not can be known by screening the presence of transgene by various methods. As we have in the vector itself, we have applied the bacterial selectable marker, plant selectable marker, and also reporter gene. So methodology to be adapted is germinating the seeds on hygromycin-resistant antibiotic containing MS medium, and performing a PCR by designing primers for the tDNA region, that is the hygromycin-resistant gene region, and also the presence of reporter gene. Also, we will prepare the primers so that the presence of the reporter gene also is an indication that the transformation process is successful. And the observations for the expression of GFP in stomata, trichomes, hypocotyl, and roots, all those things under the green fluorescent microscope we can observe. So only the transformed varieties only will emit the green fluorescence, whereas the wild type varieties or the non-transformed varieties are not appearing, not emitting any kind of the green fluorescence. That is also one confirmation. So the presence of the reporter gene itself indicates the transformation has been successful, and also the final confirmation by the presence of transgene by doing the southern blot analysis. So all these things are the methodologies to find out the transformation, that is the in-plant transformation, whether it is successful or not. So coming to the scientific relevance, so the tDNA generated transgenic using genes that can be patented, and the methodology would facilitate high throughput insertional mutagenesis of tomato and would generate tDNA insertional mutants that can be studied for functional gene knockouts of which are of tremendous importance. And also, apart from this, the tomato is eternal raw, so it is the first choice for the genetic manipulation of experiments. The standardization of the transformation of the tomato would widen the potential of the various nutritional factors input, and the in-plant transformation technology is devised to transform tomato plants, which can be extended to other plant systems also, where the flowers are big. That should be the one more criteria. And the availability of insertional lines will complement the ongoing genomic sequence progress of project of the tomato. I cut down so many the research areas. What I would like to tell is the methodology of implanted transformation, how efficient it is, and it is in comparison with the conventional agrobacterium mediated transformation by using a tissue culture lab. You can do it in vivo itself, and it is not that much laborious in comparison with the conventional tissue culture methodology. And thank you all for your time. And thank you very much, ma'am.
Thank you. Sir, what type of tomatoes will be the outcome of this experiment? What type of tomatoes will be the outcome, and what are you expecting?
Actually, ma'am, the main motto behind this technique is yes, ma'am. More kind of mutant varieties. I mean, we can so that we can design the designer based. Like for example, if you want to make an edible vaccine, so the gene of interest which is responsible for that vaccine, we can introduce in the fruit, and that can be consumed by the people. So
but again, there are more.
It is a high throughput technology, ma'am. It is a very much efficient technology in comparison with the tissue culture, so that we can produce. Yeah, constraints are there, ma'am, because it is recently